Well, hello, everyone. It's my absolute, I had to write this down because I'm very nervous. It's my absolute pleasure to introduce Jon Aguinaga. And so Jon is one of my very first PhD students. He and Kirsten, who's my other first PhD student, both started at the same time in the fall of 2020. And so when I started here as a faculty member, I was given lots of advice from other professors about how I need to be careful picking the right students. But really, they were the ones who took a leap of faith on me. I had no one in my lab. I had no track record of producing successful students, and not even a functional fisher. And despite this, he still decided to take a chance with me. And I'm so glad he did. And so Jon's accomplishments, sorry, this is going to be a little bit blunt. Jon's accomplishments are all the more impressive when you remember that Jon started his PhD during the pandemic, right, in fall of 2020. And this meant that all training had to be done either remotely or, if in person, in masks and socially distant, more or less. But even so, we still found many ways to have fun. We had lots of lab socials where we were all as socially distant as you can be in Halloween costumes with masks on. And then in summer of 2021, we, which was myself, Jon, and Kirsten, traveled up to Mendocino County to have a field season with some collaborators of mine. This was the excuse of looking at live fish in the wild and learning new field techniques. Jon has actually used a minnow trap before. But really, after a year of Zoom meetings and lots of startup to burn, it was mainly an excuse for us to explore wine country. And more importantly, visit our very favorite redwood, Old Man Dave. Since then, I've been very proud to see Jon grow into his own as a researcher, presenting his first talk at ABS in Costa Rica. And from then, he's proven himself to be a skilled communicator, as you'll see today. But perhaps was most evident to me when he could successfully get us to guess the appropriate movie title while under a sheet. Jon is also a compassionate and very talented mentor. He has mentored numerous undergrads, here's just a few, Liam and Mia, throughout his career here, participated in the Young Scholars Program for 4 years, which had him teaching a coding course to high school students and mentoring them over the summers in our lab. He's a skilled teacher too, always finding ways to break down concepts in ways that his students understand. He made this, not me. He's been a key part of our lab since the very beginning. Our lab culture now and how we treat each other is in large part because of the kindness and openness that Jon and Kirsten helped model for all future lab mates. For a long time, it was truly just the three of us. Here's our very first lab social in fall. And we built this entire lab together. Here we are now. And so Jon's going to tell you a lot about the incredible computer vision pipelines and tracking technology he's used in his work. But just know that when he started, our tracking systems looked like this. So we've improved our systems quite a bit, in no small part thanks to his efforts. And in his time here, Jon has proven himself to be just an incredible researcher. He's thoughtful, rigorous, and he's truly one of the most efficient workers I know. He's already published his first chapter, which gets memorialized on our lab door. I've never known him to miss a deadline, including this one. A neat and tidy 5 years from start to finish. Jon, I am so deeply proud of you, and I know that you have such a bright future ahead of you. Please welcome, join me in welcoming Jon.
All right. So thank you, Kate, for that intro, and thank you all for being here today. So it begins with a question. What does it mean to be social? So this is Nico Tinbergen, who got the Nobel Prize in 1973 with Konrad Lorenz and also Carl von Frisch for their observations in animal social behavior. In his 1951, The Study of Instinct, there's a particularly relevant quote, whereas an animal is called social when it strives to be in the neighborhood of its fellow members of its species when performing some or all of its instinctive activities. Tinbergen's work further spurred conversations on things like, what does it mean to be a social group? According to Ashley Ward and Michael Webster, a social group is one where two or more individuals maintain proximity in space and time through mechanisms of social attraction. These definitions have been fueled by decades of observations that animal groups are common in nature. They exist in a variety of ecosystems and span a wide range of taxa. And from these decades of work on group living, we have learned that there's many benefits to forming groups, including the ability to exploit resources from riskier microhabitats, protection against predators and intruders by means of territory defense, also reductions in movement costs through improved movement energetics in fish shoals and flocks of birds. Reproduction has also been improved while living in groups because you have easier access to mates. But particularly for me, what I am interested in is social information, how the behavioral variation within species as well as between species produces information for others to use in their decision-making. For example, you might have a meerkat that says, "Fly, you fools." What they do with that information is completely up to them. I'm also interested in the costs of group living more broadly. Maintaining memberships in groups is particularly costly. One of the more famous costs to group living is social hierarchies, and that's often depicted by the pecking order in chickens. You can also have increased conspicuousness and attractiveness to predators, as living in groups often increases the visibility to hunting predators. In 2025, I don't have to mention disease and parasite risks, so I'm just going to gloss over that one. And of course, reproductive conflicts and contests for mates who might be in high demand. But for me, I'm particularly interested in resource competition, how individuals within groups as well as between groups compete for similar resources like food, mates, habitats, and so on. From all these decades of observations on group living, we have learned that most of it has been done on single species groups. However, we also know that mixed species groups are also common in nature. And mixed species groups may form for a variety of reasons, but maybe they form to reduce the costs while simultaneously increasing the benefits to group living. And this can occur through a variety of different mechanisms. Now, the mixed species group is generally defined as a moving group of animals that forms through heterospecific social attraction. I want to shout out Darian for providing this mixed species shoal. Shout out Darian. And I want to also explicitly mention that for my talk, I'm going to be focusing in on mixed species groups that are on the move, rather than a group that is aggregated around a certain resource or clumped together, not on the move. From these observations, we have learned that mixed species groups are becoming more and more of a research interest. We have received, or have been lucky to receive, a book that comprehensively reviews the costs and benefits of mixed species grouping more broadly. We also have a pretty comprehensive review that came out in BioReviews in 2020, but most recently, there has even been a special issue devoted to the entire understanding of mixed species grouping more generally in Philtrans B. From all of these works spanning the 1960s, the first thing that I have seen is that most of this has been done on birds. People really like birds, which is awesome. I really like birds too. But the other taxa that are shown here have been far less studied. From these studies, we have learned that in general, the drivers to mixed species grouping is generally attributed to improved foraging behaviors and anti-predator behaviors. And these behaviors may be improved in mixed species groupings because species often differ in their tolerance to risk. And this can then lead to individuals from different species exploring riskier microhabitats. And in doing so, they generate increased social information for others to then use in their own decision-making. The formation and maintenance of mixed species grouping is generally attributed to these types of mechanisms, and they have been most studied in birds, mammals, and less studied in fish. So a fish shoal is defined as a group of fishes moving around in space in uncoordinated ways. And I have to remind myself to take a breather here because these next slides are, I guess, pretty important. Mixed species shoals have been a growing area of research over the last few years. In 2019, the first review about mixed species shoaling and why they exist in nature has been published in the Journal of Fish Bio, talking about whether or not the costs and benefits to single species grouping apply to mixed species shoals in similar ways. And then most recently, like 2 weeks ago, the second study was published by Ashley Ward and Michael Webster, so the person I quoted earlier, the folks I quoted earlier, talking about the mechanisms for why these shoals appear in the first place. And what we have learned is that mixed species shoals are particularly interesting to study the formation and maintenance of mixed species grouping more broadly. You could go out into nature, collect them, bring them into the lab, and I'm going to be foreshadowing here. You can particularly manipulate their risk environment using chemical cues. And by doing so, you can exacerbate the potential behavioral differences that you can observe under different situations. So we know that risk can modify the value of social information. How individuals perceive and then respond to risk influences behaviors like foraging and anti-predator behaviors. So potentially, this could be a mechanism to reduce the costs of associating with heterospecifics by increasing the benefits of behavioral variation and the information that it produces. So this then leads me to my overarching dissertation question, which is, to what extent does behavioral variation drive mixed species grouping? Now, I'm only going to be focusing on the foraging aspect of behavioral variation rather than the anti-predator aspect. So my hypothesis is that the behavioral variation exhibited in mixed species groups improves overall group foraging performance. So you might be asking, what species am I going to be working on? Well, there are two of them, right? Allow me to please introduce the amazing Amazon molly. The Amazon molly originated from a single hybridization event between the sailfin molly and the Atlantic molly, after which the Amazon molly reproduces gynogenetically, which means that she parasitizes the sperm of one of her sexually related parents to initiate the process of embryogenesis. She can parasitize the sperm from a male sailfin or a male Atlantic, but the genome, the DNA from her parents, is never incorporated into the offspring. This results in a situation that produces a clonal offspring genetically related 100% to the mom. This process has been occurring for over 100,000 years, which basically allows us to work with them in the lab at UC Davis with Kate Laskowski. So the amazing Amazon, in summary, is gynogenetic. She is a sperm parasite, usually called a sperm parasite by some folks. She's unisexual, all female, but importantly for me, she is found in mixed species shoals with either of her sexual parents in nature. So she forms mixed species shoals year long. She's producing babies year round. Perfect system for me to work with, right? Interestingly enough, we have also found that over the years, the Amazon and Atlantic molly are very similar in their ecologies. They overlap in diet, habitat and predation pressures, parasite loads, and even morphology. So how might they be different and how can they coexist in nature? Well, one potential mechanism might be behavioral differences between these two species. Maybe if they are different in behavior, they can explore different areas and peacefully coexist peacefully, right? And maybe these behavioral differences could be exacerbated by risky situations. So you might be asking yourself, I don't really buy it. Why would they behave differently? Well, clonality can play a big role in this situation. There's a lot of studies to show that relatedness definitely influences social behavior. Because the Amazon molly is a clone, she has actually been shown by Amber Makowitz and peers that she is aggressive towards non-sister clones, and she is also increasingly aggressive to heterospecifics. Well, in a mixed species shoal, you're interacting with both. So Amazons who are potentially surrounded by clones may behave differently than Amazons surrounded by a genetically heterogeneous group. So this allows me to then go on and ask for a couple of questions to address my overarching dissertation question. First, I really need to verify that our Amazon mollies are actually afraid of risk cues. So my first study is a verification study to assess, do Amazons respond to risk? Next, I address whether or not Amazons and Atlantics vary in behavior, and I test this by exacerbating different risk conditions, by testing them in different risk conditions. And then lastly, I put them all together and do the same thing again. All right, so I'm going to take you through a couple of my studies that I have done here with Kate and friends over here. So thank you everyone for being here today. So we're going to go ahead and address this first question here. Do Amazon mollies respond to risk? And here I just want to highlight that a lot of this work was done in collaboration with two high school students who came to us with the Young Scholars Program in 2022, Ishia Pasadi and Sophia Jin. Sophia is at UCLA and Ishida actually just graduated from Penn State in 3 years, just this spring. Really cool. This work has since been published last year. I'm going to go ahead and introduce the behavioral experiments to test, do our Amazons respond to risk? First, there is a standard mirror test that allows individuals to interact with a reflection of themselves. There are situations where the individual may be interacting with itself and trying to bite itself, charging against itself, but it's just a reflection. And in doing so, we can measure the amount of aggression that it exhibits in different situations. The other test that I work on is a sociability test, which is basically a metric for how much time does an individual spend with a friend or a conspecific. Lastly, each one of these individuals is assayed repeatedly, as is the nature of animal personality studies, and they are repeatedly measured in 2 risky situations, one in low risk and one in high risk. So how do we generate high risk situations? That's a great question. Well, in nature, a predator can go out and interact with a prey species. And when it interacts with the prey species, usually it's going to want to eat it. So when it tries to eat an individual prey, it's going to mechanically damage its skin. And that process produces chemical alarm cues that are then released into the immediate environment. This is also called Schreckstoff for German folks in here. And then the predator also emits its body odor into the water. Predators are pretty stinky, so it's releasing chemical cues into the water. An individual can then chemosense both of these chemicals simultaneously, and it can produce an alarm response. Alarm responses have been shown to, individuals can exhibit alarm responses to either of these chemical cues, but here we're using both of them to simulate really high danger. These fish come from an electric yellow lab cichlid. We call them Bubbles and Bobbles. So these fish electric yellow labs are providing our chemical predator odors. The Amazon molly, few generous donors gave us some of their alarm cues, and the combination of these two I will call risk cues throughout this discussion. So to address the question, do Amazon mollies respond to risk? Taking you through a few studies, a few experiments very quickly. On the x-axis here, we have the low-risk situation and the high-risk situation. The x-axes for the rest of this talk are going to be very similar, so feel free to get used to those now. And then on the y-axis, we're going to have behavior pretty much throughout the rest of the talk. So here, the first thing that I'm going to be looking at is aggression. There is no statistically significant differences between low risk and high risk in terms of aggression. However, we do see the expected response for sociability between low and high risk. This is a small shoaling fish species. So, and under risky situations, they do like to more closely associate with one another. So we do see evidence that Amazon mollies do more closely associate with their conspecific when they are in risky situations. So overall, this study allowed me to basically verify that our Amazon mollies do respond to risk. They respond to perceived threat by closely associating with conspecifics, which is a standard anti-predator response across many shoaling species. But they don't actually modify their aggression in any sort of way. So this first study is a verification study, allowed me to verify they do respond to risk which then has allowed me and a lot of my lab members to continue using this predator throughout the genesis of the Laskowski lab. So the next study that I look at, the next question that I look at is, now that I have verified that they do respond to risk, do these Amazons and Atlantic mollies vary in the responses to similar types of threat? To do this, I'm constructing an open field assay. An open field assay has generally been tested with mice and rats to test anxiety-like responses and stress. But here we are adapting it to test more different types of behaviors like exploration, space use behaviors more generally like cover use, and also metrics of performance like inspection of predators and foraging in my case. Importantly, all individual fish are tagged with a tattoo. This allows me to retain individual identity throughout the entire experiment, which then I can attribute personality differences between my species, but I'm not going to be talking about that data here. Next, individuals are placed into an arena where they're acclimating for around 5 minutes. And then after that 5-minute acclimation period, I release the fish into the arena. I initiate my Raspberry Pi. It's recording from overhead. And then with that data, I measure space use broadly, movement behavior, and foraging performance. So how does this computer vision pipeline look? Well, here's an image of that data. This situation is depicting a high-risk scenario where in the stimulus tank, we have a live cichlid predator. The individual is seen interacting with different features of its environment, including 2 foraging patches, a cover area, and of course, the stimulus tank. Importantly, this work was done in collaboration with 2 undergrad collaborators, Liam and Mia, who have since graduated and are doing their own thing. So thank you to them. Here are my sample sizes. So in the low risk situation, the treatment stimulus tank contains a conspecific. So if I am testing an Atlantic, the stimulus tank will have an Atlantic. If I'm testing an Amazon, it will have an Amazon. The medium risk situation, the individual is placed in a novel scenario completely alone in the absence of any social cues or predator cues. And comparatively, this is higher risk than when placed with a conspecific. So I'm labeling that the medium risk context. And then lastly, the high risk situation is what you see here, where the electric yellow lab cichlid is placed in the stimulus tank. They are emitting chemical cues, visual cues. This is potentially the highest risk that the individual can be placed in. And again, importantly, all fish are starved for 24 hours prior to the experiment to control for hunger and motivation to feed. And I promise they can go for 24 hours and no problem. So I won't be talking too much about the details for the computer vision pipeline, but overall, these repeated measures resulted in 534 trials, 178 hours of video data, and we trained a neural network using Sleep AI to automatically recognize the image of a fish. And then after that, we were able to attribute XY coordinates to every single frame throughout the entire trial, which then I can proofread over several months to get over to 90% accuracy. So this allows me to partition how individuals use space throughout time and allows me to address the question, to what extent do individual Amazons and Atlantic mollies differ in behavior when under variable risk? So I'm going to go ahead and go through a few metrics, including cover use. So here's the model and some information there if you're interested. The low risk situation is going to be depicted with a shaded fish to represent the conspecific. The medium risk situation is an empty tank, and the high risk situation is the cichlid predator. The behavior is going to be cover use. Boxes that contain the same letter are not statistically significant from one another. If they contain a different letter, they are. So we can see in cover use that Amazon mollies and Atlantic mollies exhibit similar cover use in low and medium risk contexts. But in high risk situations, Atlantic mollies tend to spend more time undercover than Amazon mollies. Then we have stimulus tank inspection behaviors, where in low risk situations, we see the differences between Amazon and Atlantic mollies. Atlantic mollies seem to be more sociable than Amazon mollies, meaning they spend more time closely associating with their conspecific near that stimulus tank. We don't see any differences between the two species in the medium and high risk context. Interesting to me was that the Amazon across the entire experiment was slower than the Atlantic molly in all situations. She generally explored the open space much more slowly than the Atlantic molly, potentially meaning that she's incurring more risk while out in the open. And then for foraging performance, we see that Amazon mollies forage differently in the low and medium risk contexts, but the behavioral differences disappear under high risk situations, potentially suggesting that they are conforming in these high risk situations. So in summary, what we have seen for this second experiment is that in general, the Atlantic molly is spending more time under cover than the Amazon molly, suggesting that they may be risk averse and accumulating less risk than the clonal Amazon. For tank inspection behaviors, we see significant differences between the Amazon and Atlantic molly in low risk situations where Amazons associate less with conspecifics and Atlantics associate more with conspecifics. For speed, we see that Atlantic mollies are generally more or faster than Amazon mollies, but only when measured in open space. So they are potentially darting in the open space to get into cover more quickly than Amazons who are exploring their environment a little bit more slowly. And then for foraging performance, Amazons generally spend more time in the trial foraging in low and medium risk contexts, but those differences disappear under high risk. So in general, this allows me to suggest that Amazons may be exhibiting risk-prone behaviors while the Atlantics are risk-averse. But importantly here, we see that there are differences in foraging performance, which then really gets me excited about testing these two species together in a group. Do these phenotypes extend to the group setting? That's what the next experiment is going to look at. So here's the roadmap. Amazons are risk-prone and forage more than Atlantic mollies. Now I'm excited to get into the last experiment. Do mixed species shoals exhibit improved foraging performance than single species counterparts? And to what extent do these behaviors change under risky situations? And to do this, I use a similar type of assay. It's a very similar open field assay, but rather than the cover being over here, it's now in the center. And the stimulus tank is again containing a live cichlid predator and chemical cues. In the risky situation and in the no risk situation, it is completely empty. One important difference though is that there is a cost to making an incorrect foraging choice because one of the patches contains food and the other patch does not. So my experimental system, my groups, single species Amazon groups, single species Atlantic groups, and then mixed species shoals. And they are first tested in a low risk situation repeatedly 3 times each. And then they are tested in a high risk situation 3 times each, again, after some delay, some days to rest. All fish, all shoals contain n equals 6 fish for a total of 204 individuals. And importantly, all species are tagged with a unique color. I don't need to retain individual ID, and I'll show you why in just a moment, but I do need to make sure that I know an Amazon from an Atlantic, because when the experiment's over, I don't want to contaminate our lines. So who do I use for stimulating fear in the hearts of our Amazons and Atlantic mollies? Well, allow me to introduce the Mojada tampiqueña, also known as the green Texas cichlid. All trials contain these chemical odors and predator cues that are emitted from the green cichlid. And I place all individuals into an acclimation chamber, which I don't show here, and that's on purpose. I'll show that in just a moment. After 5 minutes in the acclimation chamber, I release all individuals away from all environmental features. I let the trial continue for 20 more minutes. And then I conclude that after 20, I bring all the fish back, I put them in their own tanks. I repeatedly measure each shoal 3 times each in low and high risk contexts. A big problem in the study of grouping behaviors at least for small animals like this, is how do we keep track of individual identities? This is a big headache that I've been working on for a long time. How do we keep track of individuals throughout the trial? Well, I'm really excited to show some of the work that we've done with ID Tracker AI, which allows us to basically train an algorithm to learn individual ID during that 5-minute acclimation period. So we feed it data, and then it learns who is who using fine-scale pixel differences between each fish. And even if they are similarly sized, the algorithm can learn who is who during these 5 minutes, which is really awesome. And again, over several months, I was able to get it to be 95% accurate. So that is what this looks like here. Here's the acclimation chamber. The algorithm is learning who is who. It can attribute identity to each individual. And then that allows me to keep track of who is who in terms of species identity. This then opens up a whole bottle of types of questions that I can answer. But for today, I'll only be talking about behavioral data and foraging performance. So to address the question, do mixed species shoals exhibit better performance than single species shoals? And how does risk environment influence these behaviors? I'm going to walk you through a couple of metrics to look at this question. First, do shoals differ in space use? Again, low risk situation, empty tank on the x-axis, and high risk situation, the tank contains our Texas cichlid, Mohara tampikenia. Proportion of open space is the first metric that I will be looking at. In general, mixed species shoals exhibit lower time in open than any of the other single species compositions. But importantly, it's significantly different in the low risk situation. And this pattern is actually going to emerge often throughout the next few slides. The Amazon shoal is resembling the mixed species shoal in high risk situations. In the predator inspection situation, for the high risk, we see that mixed species shoals again resemble Amazon shoals in high risk situations, but in the low risk situation, all shoals behave similarly. For individual speed, fish that are in mixed species shoals generally travel faster than fish that are in single species shoals in the low risk situation. But again, we see that Amazon shoals, fish in Amazon shoals are resembling fish in mixed species shoals. The group center is another metric that we are interested in looking at. It measures cohesion. If all individual fish go in separate directions, the center is going to stay at the same point. So this is a metric for how the group in general is moving around. In the low risk situation, again, we see that mixed species shoals are traveling faster on average than the single species Amazon and Atlantic shoals, but they behave similarly in high risk situations, mixed in Amazons. Next, we get into the foraging performance. It appears that in low risk situations, mixed species shoals spend more time foraging, revisiting that patch to try to scrounge as much as they can from that area. But in high risk situations, all shoals behave similarly. And then interestingly, we also see that mixed species shoals deplete patches more quickly, but this is not significantly different in high risk situations. But again, Amazons and Atlantics, Amazons and mixed species shoals respond or behave similarly in this low risk situation. So I've just taken you very quickly through a couple of behaviors that we have been looking at. And of course, there's so much more to look at, including who is responsible for leading the shoal to these rich foraging patches. Oftentimes, it's significant in the low risk situation that Amazon mollies are leading the group to find this food. But this difference is not significant in the high risk situation. So generally, Amazons in low risk situations are leading the group to find the food quicker. So in short, what did we learn from this study? Well, here's a legend. So forgot to put it on here, but lavender is the mixed species shoal. The purple one is the Amazon shoal, and the teal one is the Atlantic shoal. So we can see the relationship between these different shoal types. In low risk situations, mixed shoals were slower, in low risk situations, mixed shoals were out in space less often than the Amazons and Atlantics. And this relationship changed where Amazons and mixed shoals were exhibiting similar behaviors, less than the Atlantic. Tank inspection, All shoals behave similarly in the low-risk situation, but Amazons and mixed shoals behave similarly and greater proportions than Atlantic shoals in the high-risk situation. Mixed shoals in speed were generally faster than both of the single species shoal compositions, but in the high-risk situation, they conformed. The mixed shoal and the Amazon shoals resemble each other again. So this pattern is very common throughout the rest of the talk. In centroid speed, generally mixed species shoals were faster than the Amazon and Atlantic shoals. That relationship, again, Amazons and mixed were resembling each other again, higher than Atlantics. Foraging performance, they do exhibit increased foraging performance in the low risk situation, but not in the high risk situation. Time to depletion, Amazons and mixed species shoals exhibited similar times to deplete in the low risk situation, and all shoals depleted the patch at similar times in the high risk situation. And in general, the Amazon mollies were oftentimes first to detect the rich foraging patch. So this then allows me to think about, well, why are we seeing these types of behavioral improvements in mixed species shoals compared to, say, single species shoals? It could be that mixed species shoals are performing similarly to Amazons because they are driving these behaviors. Maybe the Amazons are producing information that Atlantic mollies can then use or parasitize to improve group performance. So what have we learned so far? Well, a couple of things. Mixed species shoals outperform single species shoals in some contexts and not all. And it could be that Amazon mollies are driving these behaviors. From the individual perspective, we see that Atlantic mollies are generally risk averse, spending more time hiding, associating with conspecifics, and moving faster in the open space to get into cover more quickly, which looks like this. And we also see that Amazon mollies are exhibiting the opposite of those behaviors, spending more time in the open space, interacting less with their conspecific, and foraging more. And then at the group level, I was interested to understand whether these phenotypes expressed at the individual level would extend to the group level. And what we see is that Amazon shoals are exhibiting similar amounts of behaviors compared to mixed species shoals, which then begs the question, are they driving these behaviors? And I'm excited to continue working to see whether or not they are driving these behaviors. So rather than exhibiting an intermediate phenotype, what we have shown is that mixed species shoals and Amazon shoals behave more similarly to each other than, say, for example, an Atlantic shoal. So overall, the Amazon molly is a gynogenetic species that has been reproducing by parasitizing the sperm of her sexual parents for the last 100,000 years. When they exist in these mixed species shoals, they have to tolerate the costs of sperm parasitism because that DNA is not incorporated into the genome of the offspring. Potentially, one way to offset the costs of sperm parasitism is that the Amazons provide valuable social information for the Atlantics to then use in their own decision-making. Maybe this social information is responsible for the formation and maintenance of this particular mixed species group, and maybe that information is foraging information that is produced by the leader, the Amazon molly. So I want to thank everyone for listening. Thanks for being here today. I've got a lot of acknowledgements, but I'll just quickly go through them. Thank my family first and foremost. The entire Laskowski lab, Kate, Jiao Chen, Amin, Jay, Kirsten, Sana, Siobhan, Max, who just passed his QE today, by the way. Karen, our first lab tech, and Abby Trasinski, who is our current lab tech, who will be joining ABGG next year. I want to thank all my Pop Bio cohort. So glad to see all of you here today. The QE committee, Andy, Sebastian, Gail, Jeff, and Peter. My dissertation committee, Andy, Sebastian, Damien, and Kate. I want to thank all the young scholars that I have mentored over the years, and also the Biology Undergraduate Science Scholars Program, who was actually just defunded due to the government regulations recently. Thank you, Maddie, for taking me on as your TA during that time. I want to thank the Graduate Student Association Executive Board Council, Ben Fong, who's our president in the back, Cicely, who is also here, Luna, Doreen, Victoria, Humine, and Tamara. The public relations team specifically is the team that I've been working with for the past year. Excited to pass on my roles to Kirsten and Sarah. Thanks for being here today. All of my undergraduate collaborators, Candace Mitchell, Monse, Liam, Mia, Amelia, and Ashika. Of course, my high school students, Ethan, Nico, Sophia, Ashita, Neo. The ABGG grad group for allowing me to intrude over the last few years. The Pop Bio grad group as a whole, you guys are amazing. The Eve admin office, everyone in the Eve admin office is so wonderful. Like, we can't do anything without them, but I really want to shout out Sherry and Debbie. And I also want to shout out the Eve custodial staff. I would come to the office, have conversations with them in Spanish. It would be nice. So thank you, everyone. I want to thank all of you. I'll take any questions.
Awesome talk. Great work. I was wondering, maybe a silly question, was there a reason you used the Atlantic and not the South End as like the parent group?
Yeah, there are two reasons why I use the Amazon. First one is that this was this number of species that we had when we first started the lab. We didn't really have that many species to begin with, but I was actually interested in working with the Mexican species because the Amazon molly coexists with the Atlantic molly in northern Mexico. So I was like, yeah, I want to work with that species. But we didn't really have any, there was no like particular reason why I chose that one. We had a few numbers in the beginning, and this was the one that I'm like, I think I can really produce a lot of numbers if I were to work with this species. And we went from less than 100 to around 1,400 fish now. It's really great.
In nature, does the molly have preferences between what, like, male she parasitizes?
There's a little bit of work to look at how she associates differently with sister clones and heterospecifics. I don't know if any preferences that she would have in terms of mating interactions. It's actually, I think, the opposite, where the males are preferring to reproduce with conspecifics of their own species. And sometimes they will interact and reproduce with Amazons, but the Amazons don't go out and look for reproductive interactions. Jenny?
Talk, John.
Thanks.
I'm not sure what we're gonna do with teaching Eve 101 now that you're graduating, but that's a problem for the future. I was curious, I was trying to pay attention to this in your graphs, and it seemed like, especially for foraging, that the Amazons had much more fixed behavior across those different risk levels, and then some of the differences arose from the Atlantics maybe changing a little bit more. I could have misinterpreted that, but is that something you noticed? And do they tend to, kind of do the same thing in different scenarios more often?
So you're asking about the repeatability of the behavior for the Amazons and the Atlantics?
I don't think it's the repeatability, but that it's less sensitive to risk than the Atlantics.
Yeah, so I do think that the Amazons are maybe less sensitive to risk. They are exhibiting behaviors that are generally incurring more risk at the individual experiment, and also that it does extend to the group level. So it could be that they are less sensitive to risk. And there's a variety of explanations for why that may occur. Some of them include kin selection. So because they're clonal, maybe their genetic material continues to live on. How do they recognize that they are clonal? They do give out chemical information to individuals that they can recognize sister clones from non-sister clones. So that could potentially explain why Amazons may be more fixed in their behavior, incurring more risk than Atlantics, who are in general risk averse or shy. Anya?
Great.
Thank you.
Do you use male or female Atlantic fish and mollies, and do you think it would matter?
Yeah, I can't believe I didn't clarify that.
Maybe you did.
I definitely did not. All of my studies are with female fish. The reason why I go with female fish is because if I were to introduce males into the system, that would introduce reproductive interactions. And oftentimes when they are interacting with these males, Amazons are trying to skid away, dart around really quickly. So they would potentially detract from other behaviors that I would be interested in looking at. But I think Sana will be looking at reproductive interactions in her work.
I remember earlier in your PhD, you were assessing risk with pool noodles. How does that fit into this dissertation?
Yeah.
Or whatever happened with the.
Yes, so the pool noodle experiment, for folks who don't know, I was in Eric Sandford's SciComm class, and we created a video for that class. And part of the footage was Kirsten and I walking around in a garage in Texas with a pool noodle and scaring them that way. So that was actually our field study in 2022, the first time that we went out to the field and the last time. But what we were doing there is taking Amazons and sailfins from the wild and assessing their behavior in these pool type conditions as well. And so we collected data on behavioral differences between the two species, but then we also shipped them off to a collaborator in Florida to look at parasite load. So we use that pool noodle to simulate a large disturbance in the water. And they do respond to that pool noodle in similar ways.
I was also, you didn't talk about this in your talk, but I'm curious if you started looking at some of the personality differences, or like if you notice that there's some fish that are always the ones that lead the group to the food source, or are really scared all the time, like stuff like that.
Yeah, so for the individual level experiment, that was the second one that I looked at. I did not look at specifically personality differences between, say, Amazons and Atlantics, but the repeatability in their behavior was fairly low. So I don't know if I will look into that, but I can look into that if I want to. For the group level assay, it's actually very difficult to retain identity between trials. I can retain identity of individual fish within the trial, but I can't quite address the question of if the individual in trial 1 is behaving the same in trial 2. ID Tracker is actually releasing a update really soon, and they have messaged me just recently that they have improved the algorithm for ID transfer. So maybe if I want to, I can retrack everything. We'll see about that.
Do you think in a mixed species foraging context, the more risk-taking and potentially more aggressive Amazons might either provide a dilution effect for predators going after them instead, or potentially fend off other competitive foragers for that mixed species group?
Yeah, that's a great question. I definitely think that if an Amazon is approaching a food patch more quickly than the Atlantic molly, that it is incurring more risk than any of its group members. There's a couple of studies in social fish, goldfish, and guppies that test the order of emergence and its effect on fitness. So individuals that generally leave the safety of cover first to find food and forage in that area are often eaten first. So we can see that in those studies, at least, if you choose to leave the safety of cover first, that's often going to result in mortality. I don't test any predation contexts here. The predator is safe in an arena to its own and emitting chemical cues, but that would be really interesting to test if we can get IACUC approval for predation.
We have it.
And then, yeah, I think here.
So I know in the coral reef fish literature, there are a lot of studies that look at the effect of group size on feeding rates. And that's always usually, in my experience, focused on one species. What does the literature say about the effect of mixed schools on feeding rates? Is there real, specifically in fish now?
I'm not sure to answer that honestly, but my best guess would be that there are some species who exhibit leader dynamics in these mixed species shoals. And I would anticipate that individuals who arrive to those patches first are often going to be able to deplete them more quickly. We can actually see in some of this example that there are antagonistic or competitive interactions, aggressive interactions between the two species. They often dart and bite at each other, which potentially influence group level foraging performance. And that is actually something that I want to look at for this particular study. I didn't mention, but the overhead recording, which you see here, is actually from 2 different cameras. So I have 2 Raspberry Pis located at each foraging patch, so I can potentially look at aggressive interactions during feeding as well. I like Raspberry Pis. Grace?
Hi, good talk. It was
Thank
really
you.
nice seeing all this come together. I really find this risk adverse quality of the mollies to be really interesting. And kind of back to the other several questions, but I think it's interesting that, like, is this some sort of fitness that is being passed down since they are clonal? And like where does the line blur between, okay, risk adverse is actually advantageous to me versus, okay, now I'm actually risking my life being out here. You know, so like,
Yeah.
I don't know. I don't know if there's been like literature on that, or I just want to get your thoughts on that.
Yeah, so to make sure I understand the question, the Amazons are risk averse, or sorry, risk prone. The Atlantics are risk averse.
Oh, yes.
Is that, are you asking how beneficial that is in a group context, maybe in nature?
Yeah. And like whether, and I guess my second part of the question would be like, do you think that was a quality that is being passed down in these
Oh, I see.
progeny? Do you know?
Yeah, that's a great question. I don't know whether or not the behavior is necessarily being passed down from parents to offspring, but what we do know is that individuals who are exposed to risk early in life can translate to risk-averse behaviors later in life. And I think we also see evidence that individuals who are exposed in risk situations, the moms or the offspring that are produced from those moms are also exhibiting risk-averse phenotypes. I don't look at that particular work. Some of my lab mates do, but it would be interesting to see or explore that more closely. And I'm excited to see whether or not we address that question. Sweet. Well, thanks everyone. I'm excited. Sorry, this is the longer acknowledgence, a few minutes. I'm excited to say that I'm going to be beginning my postdoc with Patrick Green at Brown to look at aggression behavior in mantis shrimp in context dependencies situations again. This time, instead of low risk, it's going to be low and high resource abundance for burrows and things like that. And of course, I'm going to be continuing my pipelines for automated tracking. I recently went out there and helped him set up some of his stuff, and we even got one mantis shrimp tracked, which I should have shown here. So the acknowledgement side. First of all, I want to thank the Laskowski Lab. You guys have been fantastic. I've been incredibly honored and privileged to be part of the lab. We have seen so many challenges over the years. We started off with 3, me, Kirsten, and Kate. Candace came, she was our first undergrad collaborator. We have had so many socials. We went to Costa Rica together, Australia. Kate sent me to the UK. We had so many conferences together, Portland. We're going to Baltimore this coming month. But probably my most favorite memory is actually meeting Aragorn. His Google says he's 5'11", so I think that makes me 6 feet tall. I also want to thank all my friends that I have met along the way. I want to especially shout out the trivia team, Bitter Style Hilgen or Pesto Corner, for winning trivia actually a few weeks ago. My friends from the GSA, Pop Bio hikers, Khalil, who's hanging out with my dog, and all the folks who have graduated since then, including my first year mentor, Maddie and Darian, who have been very instrumental in my time here. I also want to especially shout out Carolina and Oshi. We've been through a lot together. We started in 2020, and without them too, it'd be very difficult to stand here in front of all of you today. We've gone on many hikes together. We have eaten so well. We have gone to hockey shows, and one of the things that I think they probably enjoy about me is that I take really good photos of them. And lastly, to my family, I want to thank my family for always being around. They're probably on Zoom, so thank you to them. So I regret to announce that this is the end. I'm going now. I bid you all a very fond farewell. Goodbye.